Hello students, I am teacher Svetlana and this is our second lecture on the topic of reproduction in lower and higher plants. In this lecture, we will cover the topics like development of the male gametophyte, structure of the anthropoid ovule, megasporogenesis and development of the male gametophyte. So let's get started. Development of the male gametophyte. Students, in our previous lecture, we have already seen the structure of a pollen grain. Now, this pollen grain marks the beginning of the male gametophyte. It undergoes the first mitotic division to produce bigger, naked vegetative cell and a small, thin-walled generative cell. The vegetative cell is rich in food and having irregularly shaped nucleus. The generative cell floats in the cytoplasm of the vegetative cell. Have a look at this figure here. This is the structure of your pollen grain, and then you have it divided into two parts. One is the vegetative cell and the second part is the generative cell. Now, the second mitotic division is concerned with the generative cell only, and it gives rise to two non-motile male gametes. The mitotic division of the generative cell takes place either in the pollen grain or in the pollen tube. You can have a look at this figure here, students. See, when the second mitotic division is happening, you will have the generative cell dividing into the two male gametes, and then these are traveled along with the help of a pollen tube. Now, this pollen grains are normally shedded from the anther, and when they're shedded from the anther, at this time they are in this two-celled stage, and this is common in most of the angiosperms. So basically, the development of the male gametophyte takes place in two stages. In the first mitotic division, there is a division where it produces a bigger and a naked vegetative cell and a small thin-walled generative cell. And in the second mitotic division, it involves only the generative cell, which is going to give rise to two non-motile male gametes. Students, the development of the male gametophyte actually involves this two mitotic divisions. Now, it is completed in two stages and also in two different places. Before pollination, it happens in the pollen sac, and after pollination, it happens on the stigma. That is, before pollination in the pollen sac, the protoplast of the pollen grain divides mitotically to form two unequal cells, which I already discussed. The smaller cell is your generative cell and the larger cell is the vegetative cell. Now, after the pollination on this has occurred, and when the pollen grain is deposited on the stigma, it comes in contact with the sugary fluids here, which are present on the stigma, and when it comes in contact with these sugary fluids, it absorbs this fluid, due to which the volume of the cytoplasm increases and it creates a pressure which acts on the intine, and the intine of the pollen grains comes out of the germ pores in the form of a tube, which we call as the pollen tube. So this is the pollen tube which comes out. Now, this tube nucleus has the cytoplasm and the generative cell. All this migrates into the pollen tube. The pollen tube goes down towards the ovule through the style due to chemical stimulus inside the ovary. The generative cell divides by mitosis in the pollen tube to form two haploid non-motile male gametes. So this is the second part, or the second mitotic division, where the two male gametes are produced. So eventually, you can have a look at this figure. The pollen tube travels right till the part it reaches the female germ cell, where it can release the male gametes. So this is how the development of the male gametophyte takes place. Students, now let us have a look at the female reproductive role of the flower, which is the gynoecium. It is also called as the pistil. Individual members of the gynoecium are called as carpel. And a flower with many free carpels is called as epicarpius. And when the flower has many carpels but they're fused together, then it is called as syncarpius. Now, typical carpel has three parts, which is the ovary, style, and stigma. The number of ovules in the ovary varies. For example, the paddy, wheat, and mango are uniovulated, whereas tomato and ladyfingers are multi-ovulated. So now let us have a look at the structure of anthropous ovule. Students, each ovule develops inside the ovary and is attached to the placenta by a small stalk called as furnicle. Have a look at this image here. You can see this is the placenta and this is the furnicle to which the ovule is attached. Now, the place of attachment of the furnicle with the main body of the ovule is called as the hilum. Now, this is the place where the furnicle is attached to the main body of the ovule. This place is called as the hilum. In angiosperms, the most common type of ovule is anthropois, in which the micropylar is directed downwards and is present adjacent to the furnicle or the furnicellus. Students, have a look at the diagram again. This region is called as the micropyle or the micropylar region, and here you have the furnicle. So they are quite adjacent to one another. So when the micropylar and the furnicle of the ovule are adjacent to one another, then such an ovule is called as an anthropois ovule. Now, the ovule consists of a central parenchymatous tissue, the nucellus, which is surrounded usually by two protective coverings called as integuments. This region is your nucellus and this external protective covering is called as integuments. We have the outer integument. This one is the outer integument, and then you have the second one, which is innermost, which is called as the inner integument. A narrow opening at the apex of the ovule is called as the micropylar. So this narrow opening which is left by these integuments is called as the micropylar region. Chalaza is the base of the ovule and it is directed opposite to the micropylar end. This region is called as the chalaza or the chalazal region. It is always opposite to the micropylar region. And then we have the embryo sac, that is the female gametophyte, is oval and multicellular structure and it is embedded in the nucellus. So this here, have a look here, this is your nucellus and inside the nucellus you have your embryo sac that is embedded, and this is the female gametophyte. So this is how our female gametophyte looks. So now students, let us have a look at megasporogenesis. It is a process of formation of haploid megaspore from the diploid megaspore mother cell, which is also called as MMC. This megaspore mother cell becomes distinguished in the nucellus, more or less in the center, but more towards the micropylar end of your ovule. Have a look at this image here. It gives you a brief idea about the process of megasporogenesis as well as about microsporogenesis. This we have already seen in our previous lecture, that is the formation of your pollen grains. And your megasporogenesis is concerned with the formation of the egg, which is going to act as your female gametophyte. Students, let us have a look at the development of the female gametophyte. The megaspore mother cell undergoes meiosis to form linear tetrads of haploid cells, that is megaspore. So this is your, have a look here, this is your mother cell, it's diploid. Now it undergoes meiosis to give you 4 linear haploid cells. These are those 4 linear haploid cells which is arranged into a tetrad. Now the upper 3 megaspores abort and only the lowest one towards the center of the nucleus remains functional. It acts as the first cell of the female gametophyte. Generally, one megaspore towards the center is functional megaspore. It is in fact the first cell of the female gametophyte. So this is your functional haploid megaspore. Now this undergoes three successive free nuclear mitotic division. So there is only nuclear mitotic division happening. So there's, you can see 1, 2, 3 and 4. So there are, in this you have the first, this is your starting cell where you have the first mitotic division where you have of the nucleus, which gives you 2 nuclei, then the 2 give you 4, and then the third one, the third mitotic division, gives you 8 nuclei. Now these nuclei rearrange themselves, one from each migrate towards the center, and then they are called as the polar nuclei. Moving further in the process, 3 nuclei towards the micropylar end constitute the egg apparatus. This is your micropylar end and this is called as your egg apparatus. Now it consists of large central haploid egg cell and 2 supporting haploid synergid cells. So the center one is the female gamete and is called as the egg. It's an haploid egg, and adjacent to it are the 2 synergids. Now these synergids show hair-like projections called as filiform apparatus, which guides the pollen tube towards the egg. So their role is to guide the pollen tube towards the egg. Antipodal cells are group of 3 cells present at the chalaza end. So here at the opposite end where you have the chalaza end, here you have the 3 antipodal cells. The 2 haploid polar nuclei of the large central cell fuse to form a diploid secondary nucleus or a definitive nucleus just prior to fertilization. So here you can see just prior to fertilization, they fuse to form a diploid nucleus, secondary nucleus. This 7-celled and 8-nucleated structure is called as an embryo sac. So if you look at this figure here, this is your mature embryo sac, which is called as female gametophyte. So it reaches in this stage just before fertilization. So what do you have here? We have a total of 7 cells that are present here. 6, 1, 2, 3, 4, 5, 6, 7, and we have 8 nuclei. 1, 2, 3, 4, 5, 6, 7, 8. That is the 2 in the center and 3 at each ends, which form your embryo sac. This method of embryo sac development from a single megaspore is described as monosporic development. In angiosperms, the development of the female gametophyte is endosporous, that is, it happens within the megaspore. The female gametophyte is colorless, endosporic, and is concealed in the ovule enclosed by the ovary. So students, this embryo sac is concealed in the ovule and the ovule is enclosed by the ovary. So this is how the structure of the female gametophyte as well as the entire process of development of the female gametophyte. So in this lecture we have seen the development of the female gametophyte. In the previous one we have seen the development of the male gametophyte. So furthermore we will see about how the both get together in the process of fertilizations. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning. Thank you.